This lecture is about neurotransmitter release. We're going to cover how an action potential leads to the release of neurotransmitter. We'll review the major classical neurotransmitters, that is the ones that you'll typically encounter, and then we'll go through a few unconventional transmitters. The discovery of these unconventional transmitters caused us to change the way that we think about neurotransmitters as a whole, and in general what defines a neurotransmitter. So the whole purpose of bringing our membrane potential from negative to positive and conducting that action potential down the axon is to trigger the release of neurotransmitter. So the action potential is mostly about sodium and potassium, but neurotransmitter release is all about calcium. Now, first, let's consider what is a neurotransmitter. And what constitutes a neurotransmitter has changed to some degree over the years, again with the discovery of unconventional transmitters. What seems to be pretty good criteria for me would be that neurons make it, so it can't be much of a neurotransmitter if neurons don't make it. Neurons release it in an activity-dependent manner. That doesn't mean that they all use the same calcium coming into an axon, but there should be some activity that triggers release. Finally, it needs to bind to some target. It could be on a membrane. Classically, that's what happens. It could be inside of a membrane, in the case of hormones. And it should cause some kind of an effect on that cell. Now, there are some older ideas that we had to kind of kick out of the window. For example, we used to think that neurotransmitters had to be stored in vesicles, like they are in this cartoon. And that's true for classical neurotransmitters, but not all neurotransmitters get stored in a vesicle. Some are hydrophobic. In that case, you can't put them in a vesicle. We used to think that the neurotransmitter has to diffuse across the synaptic cleft and bind to some protein on the surface of the postsynaptic site about 20 nanometers away. Well, we know that hormones can go through the membrane and then bind to intracellular targets. There's also volume transmission, where neurotransmitters can diffuse away. So again, this rule had to be thrown out. And then finally, there needs to be some clearance mechanism. Well, nitric oxide kind of broke that one because it just spontaneously degrades. Classical neurotransmitters have a transporter protein to clear it up, some enzyme to break it down, or both. Not all neurotransmitters do, though. But we'll hang out in the world of classical neurotransmission because that's mostly what the brain uses. In that case, we're going to need to put our neurotransmitter into vesicles. And those vesicles exist in two sites. They're exactly what they sound like. There's the readily releasable pool. These are found very much near the active zone. So the readily releasable pool is a pool of vesicles that can be released readily, hence the name. So these are at what we call the active zone, or the active site. And this is the area where the activity takes place. No surprise there. There's only a handful of vesicles in the readily releasable pool. I guess they should have some neurotransmitter in them. The other pool that we have is what we call the reserve pool. This is the pool of vesicles that are held in reserve. So at some site, a little bit away from the active zone, because we're not trying to release these, we got a couple hundred of vesicles in the reserve pool. So we got a pool that's readily releasable, and we got a reserve pool. This is a place for vesicles to go to right after they're born, that is recycled, get filled up with some neurotransmitter, and then once we deplete our readily releasable pool, as we start fusing some of these, we can then pull new vesicles. So this is our backup. We have a handful that we readily release, and we got a few hundred that we can then replenish that handful with. This way we always have a few vesicles docked and ready at the active zone to spit out some neurotransmitter. Now, what causes neurotransmitter release is the entry of calcium. That's what makes the active zone active. It has voltage-gated calcium channels. So not only do we have our readily releasable pool, we also have voltage-gated calcium channels that are exactly what they sound like, voltage-gated. That means what causes them to open is a change in membrane potential. In this case, it's depolarization from the action potential. So what turns them on? The action potential. What do they move? Calcium. Now you probably remember that calcium reverses at like a positive 125 millivolts or something like that, which is well above the spike of the action potential. So when the action potential arrives, that peak is somewhere about positive 30 millivolts, something like that. That means we have a full 75 millivolts of driving force to push calcium into the cell, and that is exactly what happens. When we depolarize, those voltage-gated calcium channels present at the active zone open up and calcium flows in. Too many arrows. Giving us a brief increase in intracellular calcium. That's what it's all about. If we bring in calcium, we release neurotransmitters. If we don't, we probably don't. Calcium is critical, and we can see that on the bottom in these data. So have a look at this. What it's showing you is the excitatory postsynaptic current generated by stimulating a bunch of axons and recording it from a postsynaptic cell. That dotted line there is not the reference line. That's showing you the size of the excitatory postsynaptic currents if you block voltage-gated calcium channels. In this case, with cadmium. So this is a little cation that's going to just stick into the calcium channel and prevent calcium from entering. When you do that, that normal postsynaptic depolarization that they're measuring goes away, and you get nothing. So, no calcium, no neurotransmitter release, no postsynaptic response. Alternatively, you can do some really nifty stuff, like just deliver calcium here. Let's get around all the calcium channels and just dump calcium into the presynaptic site. When you do that, you trigger neurotransmitter release in the absence of action potentials. So it's all about calcium. But hey, let's not forget, calcium can kill you. So the way that we get around this is by concentrating our calcium channels at the active zone. So it's not that the whole cell fills up with calcium, it's that we get these little calcium microdomains, little spikes, and that's shown on top in this illustration. The pinkness is showing you the amount of calcium. And what you'll notice is that it's really concentrated in those little dots that they have. Those are their calcium channels. They're showing you that not all of them open up. Everything's probabilistic, so a lot of them open up and you get these little pink domes. So little areas where the calcium concentration goes from like 0.1 micromolar to like 100 micromolar. So about 1,000-fold increase. But just right here at the active zone. So this is where I get my burst of calcium. Not so much up here, but right at the active zone. Outside of the active zone, you see a very small increase. It goes from 0.1 micromolar to 0.11. So not a big change. Definitely a change, 10% change, but that's not going to get you anywhere. So it's not that the whole terminal is filling up with calcium. If we want to do that, and we will later, we need many action potentials to overwhelm the calcium buffering system, like all the mitochondria that are present here, and allow calcium to get to extra synaptic sites. But in good old classical neurotransmission, all of the action is at the active zone. So what's this calcium doing for us, besides potentially killing us if we don't clean it up? Well, it's triggering the release of neurotransmitters. And it's doing that by affecting something called the snare complex. Okay, so let's get in a little bit more detail here. What's really going on at the active zone? Here's our active zone. We got ourselves a vesicle right here. And it's held in place by a few proteins. There's what we call V snares. These are snares on the vesicles, hence the name. Synaptobrevin with a V in it is the V snare, so that's how I remember that one. So this has an alpha helix going through the membrane, and then another one that's going to contribute to the snare complex. Then we have T snares, that is target snares, and those are embedded in the target membrane. One of these has a good T sound in it, so it's syntaxin. And again, one alpha helix for this membrane and one alpha helix for the snare complex. And then to snap all this together, we've got SNAP25. 25 because it's 25 kilodaltons inside. But this is another T snare. And again, synaptobrevin for the V snare. So collectively we have these 4 alpha helices, and I want you to think of this as an intermolecular handshake. So they're holding hands, but in this case, alpha helices. Good enough for our proteins there. So that forms our snare complex. But the snare complex isn't really that tight yet. This is a loosey-goosey, limp handshake. But we're going to tighten this up. Now the reason that it's a little loosey-goosey and limp is that there are other proteins here. Other accessory proteins. Kind of like we have accessories on our fingers. Think of these as big gaudy rings that get in the way. So if you shake someone's hand and they have a big gold ring on there or something, it kind of hurts. So that's why we have such a weak handshake. It's not that we can't grab strongly, it's that we don't want to, because we have these weird little nasty accessory proteins. Like complexin. That one's going to come up in just a little bit. So, step 1, we got to get an action potential so that we get calcium influx. So we've covered that. Now what? Well, one of 2 things, maybe both. The first thing that calcium does is bind to synaptotagmin. So this is always true. So we'll say calcium activates synaptotagmin. Tag your it. So we're tagging the synapse to release neurotransmitters. Synaptotagmin is another protein stuck into the vesicle. So let's give us a synaptotagmin. Again, tag your it. Okay, what's going on here? Well, one option, stab the membrane. So once it's bound to calcium, what synaptotagmin can do is wiggle its way into the membrane. This does a couple of things. It pulls the vesicle closer, and it destabilizes the membrane. Let me just get a quick snare complex up here, so we have that. But what happens is calcium bound, calcium right there, calcium bound synaptotagmin stabs the membrane. This destabilizes the membrane. making it much easier to break apart when the snare complex zippers up. So the membrane is incredibly stable because there's a lipid bilayer. It has a hydrophobic core that we cannot expose to water unless we dump a bunch of energy in to break apart the membrane. That's why it's a lipid bilayer. So if we're going to fuse two lipid bilayers together, we've got to break the bilayer apart, and that is not thermodynamically favorable. So what we do is stab the membrane and weaken it. This facilitates fusion between my synaptic vesicle membrane and my presynaptic terminals membrane. Because what we're trying to do is break both of these apart and snap them together, making one continuous sheet of membrane so neurotransmitters diffuse away. It's a lot easier to fuse your membranes if you destabilize them. So that's the first thing synaptotagmin does. Stab. Insert into the membrane to destabilize it. The next thing, so we're going to stab the membrane, and then we're going to stabilize the snare complex. Okay, so we destabilize the membrane. What else do we have to do? We need to get rid of our big gaudy rings. Synaptotagmin to the rescue. So not only will it cut the membrane open for us, once it binds calcium, keep in mind there's many copies of synaptotagmin, it comes over and kicks out the accessory proteins. So, step 5 here, stabilize the snare complex. In this case, we're going to remove complexin. Good. It's written on this slide. Okay, so we fire an action potential that depolarizes us, activate our voltage-gated calcium channels to give us the calcium influx. Calcium then binds to synaptotagmin, turning it on. Synaptotagmin stabs the membrane, making it easier for the two membranes to fuse. Then it stabilizes the snare complex. Once the snare complex zippers up tightly, that's going to release a whole lot of free energy. There's my lightning bolt there to drive this thermodynamically unfavorable process of fusing two membranes. Both of them have that hydrophobic core. At some point, this will be exposed to water. We have to organize the water around it. Water doesn't like to do that. That's where the snare complex comes in. Pull the vesicle closer and deliver enough free energy so we can make sure water stays happy while we break and remake our membranes there. And lo and behold, within about 200 microseconds or so of that action potential firing, we have step 6, vesicle fusion, neurotransmitter release. Once fused, that neurotransmitter can diffuse out and bind to some receptor and cause something to happen. Now, it may be all about membrane moving around. There might be some little transitional pore where actually the snare complex creates a hole in the membrane, essentially a neurotransmitter channel, where a neurotransmitter can flow out. We don't know. Again, this only takes 200 microseconds, so it's kind of hard to catch this in action. Now, of course, we're going to have to recycle. Look, we just fused this vesicle with the membrane. Two things are happening. The membrane's getting bigger, and we're running out of vesicles. We can fix both of those problems by pinching off some new membrane that's shown in the cartoon on the bottom there. Pinch off some little bits of membrane, new vesicles, and fill them back up. Just that simple. And we got to do it because we only have a finite number of vesicles. Let's say about, in this case, 270 vesicles is on the slide. We release 1 or 2 of them. every time we fire an action potential, it's not going to take long to run out. If we fire at 10 hertz, which is absolutely typical for neurons, we got about 20 seconds of life. And that's just not long enough, in my opinion. This is why we recycle, ladies and gentlemen, so that we don't run out of vesicles and our neurons can continue to communicate. We won't worry about the neurobiology of vesicle recycling, but if you really care, you know where I sit. Okay, so there's how neurotransmitter release occurs. It's all about calcium coming in, turning on synaptotagmin, and then tag. We stab the membrane and get rid of the accessory proteins so the snare complex can form. Now, normally we're going to be dealing with the neurotransmitters that we're going to cover here, the classical neurotransmitters. And almost all of them are derived from an amino acid. Others are a couple steps away from an amino acid. Your bread and butter neurotransmitters in the central nervous system are glutamate for excitation, and then GABA or glycine for inhibition. GABA is more used in the forebrain, so like the cortex where the magic happens, thalamus. Down in the brain stem and spinal cord, glycine predominates. But they're both used to some degree everywhere. Glutamate is the one and only, the greatest, excitatory neurotransmitter in the central nervous system. Now, glutamate is itself an amino acid. So is glycine. GABA, just one step away. You can turn glutamate into GABA. It's a single process. I drew the structures out for you over there in case you care about that. So we got glutamate to excite us. What do we use to make glutamate? Well, we can use another neurotransmitter, glutamine, or we can use a component of the TCA cycle, alpha ketoglutarate. So either we're robbing the cell of amino acids, or we're robbing the cell of the substrates to make ATP. What we should be getting here is that neurotransmitters ain't cheap. So the brain's going to use an awful lot of energy as a result. GABA, glutamate, they exist in equilibrium, so you can make GABA from glutamate, one-step process. You can also replenish glutamate from GABA. There's something called the GABA shunt. I've shown that's the thing down on the bottom that GABA transaminase does. Don't worry about the details, but know that these two neurotransmitters exist in equilibrium. Now, do you make GABA? It depends. Do you have glutamic acid decarboxylase? If so, you do, and you're an inhibitory neuron. If you don't express GAD, you don't make GABA, and you're excitatory. The bread and butter neurotransmitters, because they are either amino acids or very close derivatives, are cleaned up through transporter proteins. We don't see enzymatic degradation for the bread and butter neurotransmitters. We're just going to recycle these because we use these all the time. Don't break them. Just pick them up, give them a little massage, form glutamine, and then send them back over. Here we're looking at a little cartoon representation of this process. So, we release neurotransmitter. This probably deserves its own little cartoon. So we now know how we release neurotransmitter. Now what do we do to clean it up? We've made a mess. So we'll get our good old synapse up here. Got to get our readily releasable pool. And who's the lucky winner? This one right here has fused because we fired an action potential. And now we released a bunch of glutamate or GABA. I don't care which one. Doesn't matter. It's going to go over here and do something on the postsynaptic side of things. We don't care about that today, but we will very soon. We need to clean this up. Heck, let's call it GABA. This way we have an extra step here. I think I might have GABA there. Yeah, there you go. Look at that. Okay. GABA transporters are going to come to the rescue here. And one of the major players in GABA cleanup would be astrocytes. So they have GABA transporters. Cats. maybe GAT3 or something like that. Doesn't matter. They got GABA transporters. So what that's going to do is clean the GABA up by bringing it into the astrocyte, removing it then from the synapse. Okay. Once we're in there as GABA, well, if we just tried to send this back over to the neuron, it's still GABA. It's still an inhibitory neurotransmitter, and I'm not trying to inhibit this neuron. I want it to do its own thing. I want it to be happy, follow its dream. So what I'm going to do is use my good old GABA shunt, and I'll have GABA transaminase, GABA T, turn GABA into now glutamate. Heck, let's make it a square so it's a different shape. We'll then convert that glutamate to glutamine using good old glutamine synthetase. And now we've got a different amino acid, in this case glutamine, but guess what? We don't use glutamine as a neurotransmitter. So we can spit that out all day. So let's get another transporter. So we've got this amino acid transporter here that spits glutamine out, and then another transporter on the neuron to take it back in. Hey, we've got glutamine floating around here. What can we do now? Well, let's turn it into glutamate and then back into GABA. And look at that. We've recycled GABA without running the risk of stimulating any GABA receptors. This is what we call the GABA, glutamate, glutamine cycle. It's really nifty. Aside from bread and butter, let's put a little meat or cheese or fruit or something like that in the meal. Can't just have carbs and fats as much as we'd like. Another commonly used neurotransmitter is acetylcholine. Now, this is the bread and butter transmitter if you were to be an insect. And since none of you are, it's not your bread and butter. But we still use it. We use it in the peripheral nervous system as well as central. It's widely used in the peripheral nervous system to control our muscles and in the autonomic nervous system. In the brain, we have a few populations of cholinergic neurons that are pretty darned important for learning and memory, keeping us awake. So, how do we make acetylcholine? Well, this thing isn't an amino acid. But it is derived from acetyl-CoA, which is used in making ATP. So it's still expensive. Even though it's not an amino acid, it's still expensive. So how do we make good old acetylcholine? It does begin with an A, but I want it over here. So we start with an acetate, get a choline, slap them together with CHAT, choline acetyltransferase. But this isn't equilibrium, so acetylcholine can be broken down, and that's accomplished by cholinesterases. So now we actually have enzymatic regulation, and in fact that's all we have for acetylcholine. Cholinesterases, typically found at the postsynaptic membrane, are there to rapidly degrade acetylcholine back into an acetate and a choline. We'll pick that choline up back into the neuron, as the cartoon showing us here, and we'll let astrocytes clean up the acetate. No big deal. Cholinergic neurons are found in a few places in the brain, as opposed to GABAergic, glutamatergic, glycinergic. They're pretty much everywhere. Cholinergic neurons are pretty big in the periphery. Absolutely. So any lower motor neuron uses acetylcholine, preganglionic neurons, and then the postganglionic parasympathetic neurons. So in the periphery, acetylcholine is where it's at. In the central nervous system, we do find some cholinergic neurons, but they're in select places. For example, in the pons. The pons has a big collection of cholinergic neurons that help wake us up. So they're part of the reticular activating system. We also find cholinergic neurons in the forebrain, so in an area called the basal forebrain that's at the bottom of it. These are really important for learning. These die off in Alzheimer's disease, as you likely remember from last semester. Speaking of learning, we also have memory. So in the septal nucleus, or the medial septum, that's what's written over there, this releases acetylcholine into the hippocampus, exciting it and making it easier for us to learn and remember things. Monoamines are a little rarer, kind of like acetylcholine. This is a broad class of neurotransmitters. Two big distinctions we have would be the catecholamines, that includes dopamine, norepinephrine, and epinephrine, and then serotonin. As you can see from this cartoon schematic, their synthesis is pretty similar, at least until we get to something like once we get into norepinephrine and epinephrine. But both of these start with an amino acid. In the case of catecholamines, tyrosine. In the case of serotonin, tryptophan. That amino acid is then hydroxylated. That is, we add a hydroxyl group onto it. So tyrosine, hydroxylase, tyrosine, tryptophan, hydroxylase, tryptophan. Okay, we haven't made a neurotransmitter yet. We've made the precursor. So either DOPA or 5-hydroxytryptophan. Now we're going to start making neurotransmitters, and it's the same enzyme in both of those pathways. How nice. What we need to do now is remove a carboxylic acid. So the enzyme aromatic L-amino acid decarboxylase removes that carboxylic acid, giving us dopamine from DOPA and serotonin from 5-hydroxytryptophan. We're all done with serotonin. With the catecholamines, we got a little bit to do, and it's a little bit strange here. So if you want to make norepinephrine, you got to make dopamine first, and you have to then put that dopamine into a vesicle, because it's only in vesicles that we find dopamine beta hydroxylase. That's the enzyme that makes norepinephrine. So that's all done inside of vesicles. That's not so bad, though. It is kind of bad if you want to make epinephrine, because unfortunately the synthetic enzyme phenylethanolamine N-methyltransferase, it's unfortunate it has that name, but it's also unfortunate that it's in the cytoplasm. It's back out here. That means if I want to go from norepinephrine to epinephrine, I have to first release norepinephrine and then take it back up through some transporter, all before it gets degraded. So a little tricky making norepinephrine and epinephrine. The good news is very few neurons use epinephrine because it's kind of a hassle. Why do that? Epinephrine, norepinephrine, they bind to the same receptors pretty much. Just use norepinephrine. So that's mostly what we'll see in the central nervous system. Of course, we do find a little bit of adrenergic neurons in the periphery, particularly in the adrenal glands. Okay, so dopamine, even more restrictive than acetylcholine in terms of what neurons make it. You find only a couple of nuclei that have dopaminergic neurons, and they're both in the midbrain. So there's the substantia nigra that creates the nigrostriatal system, and that's shown over here. It goes from substantia nigra to the basal ganglia. Really important if you want to move. Dopamine is not only there to make us move, it also affects our mood. So that's the mesolimbic system. That goes from the ventral tegmental area to the limbic system. So structures like the nucleus accumbens to determine reward, the amygdala that plays a role in our emotion, particularly negative emotion, and the hippocampus for memory. So dopamine has some really big non-conscious effects, changing how we feel, how driven we are to do something. It also impacts the cortex as well, in this case the mesocortical. So meso in both cases is the ventral tegmental area. The mesolimbic targets the limbic system, mesocortical system targets the cortex. Here, primarily prefrontal cortex, it's dealing with some short-term memory and planning and problem solving. So it regulates our behavior. Very simply, in that nigrostriatal system, do you do something or not? but it also regulates what you choose to do via the mesocortical and mesolimbic systems as well. The noradrenergic neurons are again confined to the brainstem, but in this case, mostly in the pons. So there's a couple different nuclei in the pons. There's the locus ceruleus and the lateral tegmental area. Lateral tegmental area also extends into the medulla. But down there in the brainstem, that's where all of our noradrenergic neurons live, in the brain. We also find a population outside of the brain, right? That's listed there, postganglionic sympathetic. But if you want to get norepinephrine anywhere in the brain, it's got to come likely from the pons. And these neurons project all over. And it has similar functions to serotonin, that's why these are grouped together. Serotonin neurons are again found in the brainstem, so very much like noradrenergic neurons, in the pons, but also in the midbrain and medulla. They're in this nucleus called the raphe nucleus. And this is an enormous nucleus that extends from midbrain to pons to medulla. Norepinephrine and serotonin have similar functions in that they regulate our mood and whether we're awake or not. They also regulate pain. That's going to be a pain for you, but we will learn that. Now, monoamines have both regulatory mechanisms. They have both transporters and enzymatic degradation. Well, obviously we're going to have transporters if we're going to make epinephrine. Okay, so monoamine transporters. We have either dopamine transporters or norepinephrine transporters for the catecholamines. And don't let the name fool you. They transport both. Dopamine transporters still transport norepinephrine. Norepinephrine transporters still transport dopamine. What's in a name, I suppose? Now, these are targets of several drugs. Some of these drugs we use are prescribed, I should say, and others are drugs of recreation. Cocaine inhibits dopamine and norepinephrine transporters. This increases dopamine concentration, and remember, dopamine regulates your behavior and mood, and so, this is probably not a surprise, but cocaine makes people feel good, and it's highly addictive. It creates a very strong drive to seek it out, and we can see that over here. So, what we're looking at over here is cocaine addiction in rodents. So the time is on the x-axis. The number of times they press a little lever to get an injection of cocaine is on the y-axis. So the higher the line goes, the more they want cocaine. If you'll look on top, these are normal rats. You go pick up any old rat off the street, it'll look just like the ones on top. If you have a means of delivering them cocaine, they're going to want it. And they'll want it more than most other things. So you'll notice they keep pressing that lever repeatedly over time because cocaine makes them feel good. Bottom. The bottom is showing us dopamine transporter knockouts. That is, we got rid of dopamine transporters in these rodents. Notice there's still that upward trend. They still press the lever. Cocaine still makes them feel good. So, of course, cocaine targets both dopamine and norepinephrine transporters. Serotonin transporters are also targets of drugs. SSRIs are selective serotonin reuptake inhibitors. So they block serotonin transporters. Monoamines are also degraded. So aside from having transporters, they are also broken down by monoamine oxidase, MAO. And this is mostly intracellular, but not all of it is. It breaks down all monoamines. Whether you're a catecholamine or serotonin, doesn't care, it'll break it down. Catechol-O-methyltransferase breaks down catecholamines. That's found outside. In either case, we enzymatically modify the neurotransmitter so it becomes something else and can't bind to its receptor. Finally, let's talk about a few unconventional transmitters. And what makes them unconventional is how they get released. So neuropeptides are found at extrasynaptic sites. Rather than being at the active zone, the neuropeptides are kind of over here on the side. And they're in dense core vesicles because they're peptides, not single amino acids, but chains of multiple amino acids. And that makes them appear darker in an electron microscope. Okay, notice we're far away from the active zone. So if we want calcium to get here, one action potential will give us calcium here, and 2 action potentials here, and 3 here. So we're going to need a burst of action potentials. 1 action potential will give you your classical neurotransmitters, but if we want peptide, we need to see a burst. We'll still get classical, so let's do. But we'll also start to release neuropeptides. This is one way that neurons can communicate differences in intensity. So there's a difference between a little bit of pain and a whole lot of pain. A whole lot of pain is going to give us a burst of activity, causing the release of neuropeptides. What these neuropeptides do is bind to their receptor and cause some change to happen. What change? Devil's in the details, and those will come up later. But they always bind to G protein coupled receptors. That means they have slow and long-lived effects on the cell. Now, adenosine's a little strange in that it is co-released with other transmitters. Like, it doesn't work on its own. There's no just spitting out adenosine. There's no adenosine vesicles. It gets co-released with other neurotransmitters. So it kind of just piggybacks along for the ride. Adenosine has a couple different types of receptors. One is inhibitory, the A1 receptor, and this is what you find in the CNS. So in the cortex, for example, where the magic happens, if you'll look over here, what adenosine does is inhibit those neurons. Panel H probably tells enough of a story. So control, here they're injecting some current. As you inject positive current, you get a higher firing rate, as shown on the y-axis. If you bathe the neurons in adenosine, you'll notice you don't fire as quickly, because adenosine is inhibitory in the central nervous system. Completely different in the peripheral nervous system, and that's because it has different receptors. The A2A receptors are excitatory. I've listed GI, GS coupled, more on that in the next lecture. That's some coming attractions. So, caffeine is a commonly used adenosine receptor antagonist, meaning it prevents adenosine from stimulating its receptor. So, when you drink coffee or anything with caffeine, it prevents inhibition of the cortex. And that's how it wakes us up. It makes our cortex a little more excitable. Let's change gears a little bit and move on to something that's purely inhibitory and goes in the wrong direction. Endocannabinoids are what we call retrograde messengers. Retrograde because typically neurotransmitters communicate from pre to post. Endocannabinoids go from the postsynaptic neuron to the presynaptic neuron, usually in response to some strong input. This is just good old homeostasis, ladies and gentlemen. So endocannabinoids are derived from lipids. So we're going to cut up a little bit of the membrane. You can look at the name over there, I don't care. We just need to know that endocannabinoids are derived from lipids. That means they're a little bit hydrophobic, and so you can't put them in a vesicle. This is why we had to kind of throw out that whole vesicular release thing. We're not going to have little vesicles of endocannabinoids in the dendrites or anything like that. No, when the dendrites are strongly stimulated, that causes them to activate the enzymes needed to create endocannabinoids. And when we do that, endocannabinoids, being a little hydrophobic, can move right through the membrane and go to the presynaptic site, stimulating their cannabinoid receptors. And these are inhibitory, meaning they're going to inhibit release from the presynaptic site. That's their job. So if you get strongly excited, tell the presynaptic site to tone it down a little bit. What's shown on top here is the effect of a synthetic cannabinoid, so something that stimulates cannabinoid receptors, presumably. And we're looking at the amplitude of the postsynaptic current. In other words, about how much neurotransmitter is spat out. And what you'll notice is that over time, those little dots start to coast downward, meaning the postsynaptic current weakens. And that's because we're just not spitting out enough neurotransmitter. But hey, on the bottom, if you inhibit the cannabinoid receptor, the CB1 receptor in this case, that cannabinoid doesn't do anything anymore. This is just a little control to tell us, hey, this is acting through the cannabinoid receptors. When you stimulate cannabinoid receptors, you are less likely to release neurotransmitter. Now, remember, fire together, wire together, that's how we learn. If you're the presynaptic neuron firing a bunch, but not spitting out a whole lot of neurotransmitter, you're not likely to fire together with the postsynaptic neuron. And this is probably how endocannabinoids interfere with memory. So I've heard. Nitric oxide. Okay, let's change gears again and go back to excitation. So nitric oxide is kind of like endocannabinoids, except it's excitatory, not inhibitory. But you can't store it in a vesicle, and it's made on demand. And it's also a little hydrophobic, so it can go right through membranes. What's a little different here is that there's no enzymatic degradation, like with endocannabinoids. Well, we can break them down. Nitric oxide just spontaneously falls apart in about 30 seconds. So when a neuron is excited, it gets a lot of calcium influx that stimulates neuronal nitric oxide synthase, and it spits out nitric oxide in all directions, affecting any cell in the area for the next roughly 30 seconds or so. The effect of nitric oxide is to promote presynaptic release. So whereas endocannabinoids decrease release, nitric oxide increases it, and we can see here. So control, the no treatment on top, they're just showing you that their acetylcholine measurements are pretty stable over time. So they're looking at 2.5-ish hours, and they're just measuring how much acetylcholine is there. If you don't give any drugs, it doesn't change that much. What's more interesting are the filled circles. When you decrease the production of nitric oxide by using that nitric oxide synthase antagonist, NLA, notice what happens. Black dots kind of creep downward. We're releasing less acetylcholine because we don't have as much nitric oxide around. On the other hand, put on a nitric oxide donor, so if you put something in the bath that increases nitric oxide levels, that's that SYN1, notice black dots peak. And we're releasing now, hey, almost twice as much acetylcholine as normal. Remove the drug, and it comes back down closer to normal levels. Now, what's the effect of these neurotransmitters? Well, that's the story for next lecture. For now, if you have any questions about how we release neurotransmitters, or kind of the general, how do we make them, how do we break them down, you know what to do. Fill out that questions box, and I'll get back to you when I can. See you later.